A viral claim promises a 90-day mushroom challenge that rewired a human brain, shocking doctors with scans said to show the impossible. But no one can trace the original post, and every neuroscientist knows MRI images can fool even experts. Could lion's mane really regrow your mind, or is the story hiding a far stranger explanation? The search for answers begins right at the heart of the hype. Searches for the original post behind the 90-day lion's mane brain scan claim lead nowhere. There is no named patient, no clinic, no date, only a chain of blurry screenshots and recycled captions. The so-called evidence surfaces on TikTok, Reddit, and Instagram, each time stripped of context with side-by-side images and the same punchline. Doctors said it was impossible. Every attempt to track down the source ends in dead links or reposts. Not a single video or medical record reveals who was scanned, what machine was used, or what protocols were followed. The word impossible takes on a life of its own. In medical notes, doctors sometimes say impossible out of surprise, not as a scientific verdict. A patient may recover faster than expected, and a clinician might say that it defies their expectations, not the laws of biology. Online, the word is transformed into a promise of miracles. What remains is a screenshot, not a scan, a caption, not a case study. There are no raw imaging files, no radiologist report, and no peer-reviewed publication. Only viral fragments multiply with every share. The mystery deepens. If the story cannot be traced, what exactly is being shown? Before the science of mushrooms or the language of MRI, the real question is whether the claim itself has any foundation at all. The need for hard evidence becomes urgent. The search for truth moves from social media hype to the standards of real investigation. To evaluate an extraordinary claim, we need raw scans, consistent imaging protocols, blinded radiology reads, and clear clinical context. Without those elements, dramatic before and after images are only images, not proof. Lion's mane is not just a culinary curiosity. In labs, it is known as Hericium erinaceus, a mushroom packed with compounds called hericinones and erinacines. These molecules caught scientists' attention for one reason. In rodent experiments, they trigger a surge in nerve growth factor, NGF, a protein vital for the survival and growth of neurons. When mice are fed lion's mane extracts, some studies report sharper memory and faster maze solving. The effect seems to come from NGF and sometimes BDNF, the brain's own growth signals, ramping up in response to these mushroom compounds. In aging or injured animals, lion's mane has even reduced the buildup of amyloid plaques and calmed inflammation markers like iNOS and T-bars. This molecular story is seductive. If a mushroom can boost brain chemicals in a mouse, could it do the same in people? The leap from petri dish to human brain is a long one, but the biological rationale is hard to dismiss outright. The idea of a natural supplement nudging the brain's repair systems feels promising, and it fuels hope and headlines. Even without dramatic human data, the pathways look plausible on paper. That is why lion's mane keeps showing up in wellness circles as brain food. The science is early, the evidence mixed, but the mushroom's reputation for supporting nerve growth has roots in real molecular signals. The next question is whether these signals translate into measurable benefits for people, or if the story fades outside the lab. A closer look at the clinical trials reveals a story far less dramatic than the viral headlines. In the most widely cited study, researchers in Japan enrolled 30 adults with mild cognitive impairment and gave them lion's mane powder, 3 grams per day, for 16 weeks. Their scores on the Hasegawa dementia scale improved during the supplement period, but within 4 weeks of stopping those gains faded. The effect, while real, proved temporary and modest. No lasting transformation, no reversal of disease. Another trial in menopausal women swapped capsules for cookies. After 4 weeks, those eating lion's mane reported lower anxiety and lower depression scores compared to the placebo group. These changes were measured with questionnaires, not brain scans, and the study involved just 30 participants. The benefits, if any, were limited to mood and subjective feelings, not structural changes. A more recent pilot in young, healthy adults tested both acute and chronic effects. After a single dose, some participants performed faster on the Stroop task, a test of mental speed. But after 28 days, cognitive scores were mixed. Some measures, including delayed word recall, actually fell below placebo. Any improvement in stress or mood was small, and the sample size, just over 40 people, left more questions than answers. Across these studies, the pattern repeats: small groups, short durations, and effects that rarely outlast the supplement. There were no MRI scans, no evidence of new brain tissue, and no doctor declaring a result impossible. The strongest claims dissolve under careful research. A clinical trial researcher sums it up: promising signals, but nothing miraculous. A neuroradiologist sits in a dimly lit reading room, scrolling through a series of anonymized brain scans. To the trained eye, what looks like dramatic change is often just a technical sleight of hand. MRI images are not photographs. They are reconstructions built from data slices collected in 3 dimensions. Shift the angle of a scan by 10 degrees, and a brain region can appear to swell or shrink by as much as 20%. Even a slightly tilted headrest or a new software update can produce images that seem to defy biology. Test-retest studies in healthy adults show that brain volume measurements can fluctuate by up to 3%, even when nothing has changed except the scanner settings. Automated segmentation tools, the kind used in many clinics and research labs, introduce their own noise. One neuroradiologist says, "We see these artifacts all the time: different slice angles, hydration levels, even time of day. If you do not control for every variable, you can manufacture a miracle." In the adult brain, true structural change is slow and subtle. Over 90 days, natural variation is measured in millimeters, not dramatic leaps. Most published claims of regrown tissue collapse under careful review. Swelling from inflammation can resolve and make a scan look fuller. A night of poor sleep or dehydration can make the cortex appear thinner. Without strict protocols, same scanner, same sequences, same positioning, before and after images become unreliable witnesses. The illusion of regrowth is easy to create, but almost impossible to trust. Running a credible 90-day supplement experiment means controlling every possible variable. The starting point is a detailed baseline. Log sleep hours, caffeine intake, exercise routines, diet, and stress levels for at least 1 week before you begin. Use the same cognitive tests at the same time of day for each assessment, and rotate between alternate forms to prevent learning effects. Consistency in timing and test version reduces practice effects and expectation bias. If you include brain imaging, schedule every scan at the same facility, on the same machine, with identical settings and positioning. The radiologist reading the images should be blinded to the scan dates so comparisons are unbiased. Avoid adding new habits or changing routines mid-experiment, since even small lifestyle shifts can sway results. The goal is to isolate the effect of lion's mane, not to stack it with other changes. Only by holding these factors steady can any improvement or lack of improvement be plausibly traced back to the supplement itself. Treat this as a small study and let the evidence speak. 3 explanations stand out. The most likely explanation is that the scan images were never truly comparable. Small changes in angle, software, or hydration can turn a routine follow-up into something that looks extraordinary, but is not. Next, lifestyle shifts often arrive alongside new supplement routines. Better sleep, less stress, and more exercise can each sharpen memory and mood, with or without a mushroom capsule. Finally, transient changes like reduced inflammation or improved hydration can subtly alter brain appearance on MRI, creating the illusion of new growth. None of these require magic or miracles. They are the known quirks of brain imaging and human behavior, amplified by algorithms hungry for viral stories. Today, viral health claims travel faster than peer review. With human brain science, what is plausible matters more than what is popular. Until evidence matches the headlines, skepticism protects us better than hype ever will. The real frontier is not miracle scans. It is learning to question what we hope is true. Share your perspective below.